Okay. Welcome everyone who's joined this lab tour. We have got Jake and Jonathan here who are willing to show us around the lab tour, around the lab. Jake has done his undergraduate at Durham, Jonathan hasn't, but they've both got very interesting things to tell them about their research here. So I'm going to hand over to Jake now and enjoy the tour. Oh, and feel free to turn on your camera or to unmute yourself when you've got any questions. It's supposed to be an interactive tour instead of having to wait to ask your question until the end. So feel free to interact anytime during the tour. Yeah,
okay.
over to you, Jake.
Okay, so I'm Jake, and oh, the lights have just gone off. What I'm going to be telling you about today is our work on rubidium cesium molecules, and I'm going to turn my camera around so you can see what I'm looking at. So the rubidium cesium experiment works in 3 stages. We start with laser cooling of atoms down to about 100 or a little bit more than 100 microkelvin. So our first stage is really getting down to a few millionths of a degree above absolute zero. So we're all when we say we're talking about something hot, we're already talking about something that's colder than the deepest, darkest regions of space. So this lab is one of the coldest places in the universe. But yes, that's our starting point. We start with a very, very, very cold gas of rubidium and cesium atoms. We then do a process that's known as magnetoassociation. And what magnetoassociation refers to is we start on some energy level that's somewhere over here. And then by using a magnetic field, we can bring these pairs of atoms such that the minimum of energy lies when they're some distance apart. In this case, the distance is something like a nanometer, but they're sat very, very close together. But in terms of a molecule, this is still very, very weakly bound. They've only got a few kilohertz of energy. So any stray radio waves, any stray microwaves that are in the lab from maybe an overactive Wi-Fi router or something, they can break this molecule apart, and we can lose it. So this leads us into the final stage of our experiment, which is it's called STIRAP, and it's effectively just a 2-color optical process that forces the molecules from, I don't know if you can see this very well, but from this very, very highly excited state via some even more highly excited states to a very, very tightly bound ground state. And this difference in energy here corresponds to something like a few hundred to a few thousand Kelvin of thermal energy. So not only have we got a gas that's very, very cold, we have a gas that now can't break itself apart. Once we've done that, we've done lots of interesting work with the molecule gas itself. We've looked at how the molecule collide together and what happens. So we're looking at chemistry, just a few billions and millions of a degree above absolute zero, which is very different when your previous experience of chemistry is with a Bunsen burner and a test tube. So you're completely probing a different parameter space entirely. We've also tried to explore the internal structure of the molecule. Because the molecule is made up of 2 separate atoms at these ultra-cold temperatures, the effect of hyperfine structure is massive. So you can really look at nuclear interactions in this system. So that's kind of the broad overview of what we do. But I'm now going to show you inside and what that actually looks like, because it doesn't look as nice as the pictures. So our experiment is broken up into 3 main tables. We have a table here. Hopefully I can turn it around. So we have a table here, which is entirely dedicated to generating light used for the laser cooling. We have a second table for lasers, and this is entirely dedicated to light for the molecule STIRAP process. And then we have a third table, which is currently boxed up, which is entirely for holding our whole atom. But I'll walk you through what each of them do. Again, as Kiki said, please interrupt me and ask questions if you have anything. I'm very willing to answer questions. So if we take this laser cooling table, what is it? So for each species, so we have two. So we split the table down the middle. This side here is for the rubidium. This side here is for the cesium. Because they're different elements, they have optical transitions at different wavelengths. They have different colors of light that they need to do the specific excitations that we need for our experiments. In our case, this left-hand side works at a wavelength of 718 nanometers. The right side works at 852 nanometers. So they're both firmly into the infrared. So we'll just take the rubidium side for an example. What we need to do is we need to take the light that comes out of a laser diode. So inside this metal box here, there's a laser diode and a diffraction grating to select off one single wavelength from that laser diode. What we need to do is we need to take that light, and then we need to manipulate it to be not only in the right frequency state that we need for our experiment, but also to be in the right polarization state. So if we follow through some of these optics, and as you can see, there's quite a few of them, so this is a bit of a pain to set up. But if we take some of these optics, we have to pass them through various components that slightly change the wavelength of the light by a few, well, change the frequency of the light by a few tens to hundreds of megahertz, and rotate the polarization of light by a specified amount that we're choosing, so that we can create this light that is suitable for laser cooling. I believe Simon talked about laser cooling a little bit in his talk, but that's just helpful if I just give you a little refresher. The idea with laser cooling is that when an atom is moving in a specific direction towards a laser beam, it gets Doppler shifted onto resonance. So what we need to do is we need to compare our laser light on the table to atomic samples, so that we can make sure that we're not on resonance, that we're a very specific amount, what we would say detuned from resonance. So even though rubidium and cesium are metals, and they are alkali metals, if you want to relive the early 2000s, you should have a look for videos of Brainiac throwing rubidium and cesium into a bathtub. At room temperature, they're very willing to form gas. They have a very low vapor pressure. And so what we actually only need is to make for our samples is what's known as cells, these tiny little glass test tubes, it's probably the best word for them, that are filled with a very small amount of rubidium metal or cesium metal, and then allowed to create vapor that we can then probe with our experiment. So all of this system allows us to generate 6 beams for each species, which are a very specific wavelength for the laser cooling, and so that we can, as the atoms are moving towards them, they get a kick in the opposite direction and get slowed down. We take this light over from this table, so this is the same, this table here is the same one you've just been seeing on the other side. So we take this light through some optical fibers that run through this little piece of plastic, and they go onto the main canopy. This main canopy has a vacuum system inside, so just some huge pieces of steel and glass. And within there is a large amount of rubidium vapor that we can access and use to make samples of gas that are at ultra-cold temperatures. Are you running a shot now, or? Okay. So in a moment, what I can show you is what it looks like. So the main element of our chamber is this pair of coils here. And what is between them is which you might be able to see actually, because you're in a unique position in that I can actually manipulate what you can see. So you might just be able to see between those two plates, it's a bit dark, there's a small glass cell, and it's exactly the same as the cells I showed you on the other table, except it's held at a higher vacuum. So there's less air in there and more rubidium and cesium vapor. So the interactions we're looking at are only of the rubidium and cesium. Around the cell are these two pieces, these two green things. I think they're appearing on my camera. These are just some fiberglass mounts to hold some magnetic field coils. So just some pieces of standard copper wire that we wound into circles. We use those to generate, by passing like a 200 amps through them, we use those to generate the magnetic fields for the MOT. So this magnetic field gradient that lets you access specific atoms in specific directions. Also for generating the magnetic field for sticking the atoms together into molecules. The final stage of I'll skip ahead a little bit from the early stage of our experiment. So the final stage of our experiment is when we need to look at the atoms. Looking at atoms at ultra cold temperatures is a little bit different to how you might imagine looking at, say, stars or other bright objects, because they don't actually emit any light on their own. So what we use is the fact that these atoms will absorb light to cast a shadow onto something, and we then look at the shadow and how dark that shadow is to generate an image. It's so-called absorption imaging. And we do that with just it's just a very fancy but fairly standard CCD camera, which works exactly the same as the one in your phone or your webcam maybe, but it's just well, it's probably about £30,000 more expensive than the one in your phone. So I think, are you about to run the experiment? Okay. So the sum total of all of that is that we can generate gases of atoms that are at ultra cold temperatures. And if you look at that in the infrared, it looks something like this. So this little bright spot here is a cloud of rubidium-87 atoms. This little thing is just a reflection, probably, that are being cooled down and held within a magnetic well, constantly being cycled by light to keep them at a few hundred microkelvin, or less than a few hundred microkelvin. As we then go on, we can then make them move by changing the magnetic field. So you can see that they jump because the magnetic field changes. And when we turn off the lights, they should all just disappear because, and this smaller one that you saw briefly, that was just the second species in our experiment, that's the cesium. We don't need as much of that, so we keep it quite small. Okay, so that's a very brief overview of our experiment. Does anyone have any questions that they'd like me to answer whilst I'm here?
I've got some questions.
Yeah, that's great.
So on the first table you showed, there was like a lot of different, like a very complicated setup, basically.
Yeah.
Do you need to like change that setup to do different experiments, or is that just do it once and then don't touch it ever again?
So for our experiment, we don't have to touch this very much. We've had to do some changes to it if we need a new color of light for whatever reason. So let's say we want to look at the atoms at a higher magnetic field, or we want to look at I don't know, pushing the atoms in a specific direction for some reason. We need a new color of light, so we need to find space and then put in all the optics we need to make that new color of light. In other experiments in Durham, so I think maybe the one Jonathan's going to show you, where you have lots of isotopes, you need to change the setup more regularly because each isotope also has a specific set of transitions, not just each atom. And so to get those different colors, you need to make very, very small changes. Don't know if that's really answered your question. It's a bit hand wavy, but the answer is mostly no, but sometimes yes.
Okay, thank you. Do you know like how long it took to set up to do the initial setup?
So I don't know how long it took the first PhD student to do the initial setup. I do know that the first two PhD students, one of theirs was on making the MOT. So the thing I've just showed you, that was about 3 years from going from an empty room to going to having an ultra-cold gas. It can be quite time consuming, and in these early stages, there's lots of things that can go wrong. So you end up having to start again a few times. But I think it's it was at least a year, if not 3, to go from an empty room to a fully working experiment.
Thank you.
Is there anyone else? Okay.
Sorry. Hi. What are you currently looking at? Are you looking at like the transitions when they're very cold?
Yeah. So what we're looking at the moment is we're looking at the transitions inside the molecule. So the advantage we have of the molecule system being very cold is that we can address individual quantum states in the molecule. So when you do traditional chemistry spectroscopy, even at something like 7 or less Kelvin, which is a pretty standard temperature for chemists, the spread of the energy of the molecules just from moving around means that the rotational hyperfine and sometimes even the vibrations of the molecular bond blur out all of the different quantum states. So you end up with this kind of like quasi-continuum of states. Because we're so much colder, because we're just a millionth of a degree, the energy the molecules have from moving around isn't enough to blur those states out. So we can probe the quantum mechanics of the molecule. We've also, in the last year, looked at what happens when 2 molecules collide, trying to figure out what kind of process is going on, whether it's chemical, whether it's physical, whether there's something more complicated going on. I can't really explain that to you without having to show you some diagrams, unfortunately. If you are interested in that, I can send you some papers and I can give you an explanation over email, if that's okay.
Yeah, I think that's answered well.
Hi, I've got a quick question, if that's right.
Yeah, that's fine.
So you mentioned in terms of like people previously doing a very similar PhD, like setting up the equipment and then you're kind of now using it, or you have used it for your PhD. Is that quite common, you think, for PhDs, like to kind of follow on from other PhD work? Yeah,
I think it's a classic case of, in research, if you see failure, it's because you stood on the shoulders of giants. No PhD exists in isolation. I mean, maybe some PhDs exist in social isolation at the moment, but the main thing that a PhD student will do is they'll have like one specific problem to solve. So that might be making a MOT, that might be making a gas of very, very cold rubidium or something. The next PhD student will then have another job or another problem to solve, and that problem might be sticking together rubidium and cesium atoms to make a molecule. It might be finding some molecular transitions. it might be controlling a single state of the molecule, and it might be looking at collisions. As it goes, those are the last 5 or so PhDs in our lab, so they're quite easy to name. But you very rarely have someone who goes into a lab and then having something already there and then tears it all apart and starts again. Mostly because it's just not very efficient. Someone's already worked really hard to build something. You don't really want to go in and destroy 3 years of their work just because you think you might do it a bit better. That's not to say that doesn't happen. Sometimes, new pieces of equipment come along that just work better. Like sometimes, for instance, on this table, that I'm stood by. So we have a mix of these metal boxes with the yellow writing on. Some PhD student at some point has made these. These are homemade and handmade. But on the far side, you might just be able to see there's a little blue box. Maybe I'll go down a little bit. So there's these blue ones. Those have been bought. So when we had a little bit more money, someone has decided that the home-built laser just doesn't work well enough anymore, and we needed to replace it with one that a company makes. And that's more likely that what you'll find is rather than replacing something, you'll upgrade it. So something that someone's worked hard on, you'll replace with something that you can buy, only if it does something better. Does that help answer your question a little bit, Josh?
Yeah, that was really interesting. It's just not something I've really thought about before. Yeah, it's really interesting. Thank you.
I guess I have a question as well.
Yes.
I imagine that this lab will be taking on new PhD students this year. What sort of, I guess, little problems will those people end up tackling?
Okay, so we've just taken on a new PhD student. And his project is looking at how we can use the techniques that I've developed in terms of quantum control to do something useful. To put it bluntly, there's this big idea in quantum technology at the moment called quantum simulation, where you use one quantum system that looks a little bit like the thing you're interested in, and you try and use the one that you have control over to learn something about the one you don't have control over. So most often this is used in reference to cold atom systems and condensed matter systems. So condensed matter systems are horribly complicated because you don't really have control over where the atoms go and how the atoms interact. In cold atom systems like mine, or like the communities, you have control over where the atoms go. You have control over how they interact. So what you can do is you can make your cold atom system behave like the condensed matter system, and then you can figure out what's going on inside the system that you have no control over. So our new PhD student is going to be trying to figure out how to do that using the techniques that we've developed. He'll probably have to develop some techniques of his own, but he will have a lot of help and experience in me, and another former student will be helping him along with that. So he won't be starting from scratch and won't have to be discovering everything for himself. On top of that, when you work in a lab like this, there's lots of little problems that you can't foresee. So I don't know, maybe one day he'll come in and I don't know, a fuse will have blown in a piece of equipment that no one's looked at for 10 years, and he'll have to figure out how to replace it. But in terms of science questions, yes, he's going to be looking at using these molecules as a quantum simulator. Does that help answer your question?
Yeah, thanks. That's really interesting to sort of know where the lab is at the moment and where it's heading. Yeah, perfect. Yeah. Thank you.
I should also add to anyone else who's listening to this, maybe when it's been recorded and put online, if you have any questions, about what the lab's doing, anything else you want to know about, feel free to email me. I'll give my email to your president so that you can forward stuff on. Yeah, I'm always happy to answer questions if anything comes up.
Good.
Okay, I think if that's everything, I should hand over to Jonathan, who's going to be showing you our other lab.
Yeah. Okay. Can you hear me okay?
I can certainly hear you, Jonathan. So.
I was wondering, I might share a PowerPoint just to, in lieu of the poster. So we don't have a poster yet, but. So I work in a lab that is kind of building on the work that is done in Jake's lab. And we're going to build a cold atom machine that's able to make rubidium cesium molecules. And then we're going to be using this technique that's been recently developed called quantum gas microscopy. So in this, we take atoms which we have, and they're very cold, and we put them in something called an optical lattice, which is basically a sinusoidal potential of lots of little wells which atoms can sit. And it's very similar to maybe the simplest idea of a crystal from your condensed matter course. You'll think about electrons in a crystal and block waves and those kind of things. And as Jake was saying, there's this idea of quantum simulation. So these atoms moving in optical lattices are very similar to electrons moving in a crystal of silicon or something. But the advantage we have is we can control and cool down these atoms on a single site level. So here people have been drawing pictures in their clouds of atoms by raising the potential on certain sites. And what we're going to hopefully do at the end of the day is to have molecules in such a lattice. And these molecules can interact very strongly and simulate or show the dynamics which are similar to strongly correlated condensed matter systems. So that's where systems where the interaction between the particles is very important to the dynamics and what goes on. Okay, so I'll stop screen sharing and hopefully show you around the lab on my phone now. Okay, so hopefully you should be able to see the lab. Okay, so I'll start from the laser table, the cooling table. So we have 4 tables in our lab.
I'm not sure if it's just me, but I can't really hear you very well.
Eventually we'll have the lasers which combine atoms into molecules. Then over here we have our cooling lasers. So this is very similar to the setup Jake was showing you with the home-made lasers. Except we had slightly more money at the point when we started this experiment, so we could buy all our lasers from Toptica, which is a German company that make lasers. So again, very similar idea to Jake's experiment, where we take light from a laser diode and send it through optical fibers, reference it to a cell containing rubidium. So that's a kind of a way of measuring the frequency based on some physical constant, like the transition in rubidium. And then we use these beam splitter cubes to divert it around and control the polarization. And these devices called acousto-optic modulators. And basically this just is a diffraction grating which is controlled by a sound wave in a crystal. And the combination of the sound wave and the optical wave changes the frequency of the laser as it goes through this crystal. And yeah, that's the table. And all this is, as Jake was saying, is light to control our very cold atoms. So again, feel free to interrupt me. I can hopefully hear you on the phone. Okay, so we collect all the fibers up into this bundle here. Maybe you can see that. And then they get sent over the top of the lab down over here, which is where we have our vacuum chamber. So hopefully you can see there is this big steel box with like portholes with viewports on the side. And that's got three things coming out of it. So it has two cells on each side where we prepare our cold atoms. So the atoms start their life, as it were, in a little dispenser here, which just emits very cold vapor, or not very cold vapor, sorry. It just emits room temperature vapor into the vacuum chamber. And then in this cell, we do the initial cooling, where we take atoms from room temperature and then take them to about 1/1000th of a degree above zero using this Doppler cooling effect, where only the atoms moving towards the laser will get a kick from that laser beam, and so the kinetic energy reduces. And then they get sent into the main chamber here, which is where we do most of our science at the moment. And the plan after that is to move them along into a special glass cell, which I can maybe is hiding under a tube at the moment to keep it from people's grubby fingers like mine. But maybe you can see it there. And that glass cell is just allowing us to get a bit more room for our lasers and our lenses around the atoms. And specifically, it allows us more room for our microscope objective, which is being tested at the moment over here. So you have to imagine what it will look like when it's underneath the glass cell. But the idea is that using this high numerical aperture, so very wide lens, we can collect lots of light from the atoms as they fluoresce. So you'd imagine that the atoms would be sitting back here, and we would collect the light from using this lens and then focus it on a camera, which would hopefully allow us this kind of single site resolved imaging where we can see atoms on individual lattice sites of this optical crystal. Yeah, that's the basic idea behind our experiment. I could probably take any questions you had now, or see, can we get the camera back to me?
Hey, I was wondering, the tables are so much more denser than I thought in terms of components. Do you have an estimate for like how far this laser beam will travel within the table? It seems like it'll be a very long way.
That's a good question. Never really thought of that. I think it's usually around like a quarter of 1 meter or a few meters. And I guess the important thing is that we try and use laser beams which are wide enough that they won't diverge much over that path. So maybe it's a bit counterintuitive, but the tighter you focus a laser beam, the faster it will spread out. Maybe I could draw a wee picture. But the basic idea is like, as long as the laser beam isn't too tightly focused, we could just send it as far as we liked and all over the place. But I guess like this kind of thing would be happening. Trying to, here we go. Nope, I did it too many times. Yeah, so this would be a tightly focused beam, whereas for a wide beam could travel very far with f-diamond. Yeah, that's a fun question. Thanks, Tom. Any other questions?
Hi. I guess, you know, same as last time, where is this experiment sort of going, like in the next year or so? Like
Good
what's...
So in some ways, we're a bit of a baby experiment compared to, Jake's lab. So we're still working on setting up lasers, and at the moment we are setting up the lasers we'll need to make this optical crystal or an optical lattice. So we have these guys over here, which are like very high power but far detuned infrared lasers. And the hope is that once we've got those all set up, we can transport our atoms, which is like not the easiest thing to do once you've got a very delicate sample of very, very cold atoms. We'll transport them from this big cell chamber into the small cell and set up that microscope objective underneath them. So I think, like, realistically, we won't have the next aim on the experiment ultimately is to get some single site resolved images of atoms. That's probably realistically the goal of my PhD. And then the person afterwards would be moving on to setting up for combining those atoms into molecules. And then, and like this task is maybe like probably at the cutting edge of what in cold atoms at the moment. So us, a few groups, one from Princeton, one in University of Boulder, Colorado, and a few others are working on this problem of molecules in an optical lattice with single site resolved imaging control. So that's where we're heading.
Yeah, thanks. That's really interesting. Yeah.
Good. Yeah, and in case it isn't obvious, we're, well, maybe it's sort of like we're recruiting for new PhD students, and yeah, so it's like definitely get in touch with Simon if you're interested in these kind of things. I think there's information on the website for the on the physics department website, QLM.
What do you think has been the most interesting or rewarding about your research so far?
It's a good question. Very optimistic. I think like just seeing things in the flesh and like in working in real life. So before this, I've done like a theoretical master's project because the lab in the university where I was basically lost the vacuum in their vacuum chamber, so they couldn't do any cold atoms for about 6 months until they got the vacuum back. So seeing like real cold atoms and being able to make progress on that's been good. I've also really enjoyed it's like the kind of, I guess, being part of this academic community. So like going to conferences, hearing other people's talks, gradually learning more about what's interesting at the moment, what people have done before, and like, that's been really good too. Get to read some really interesting physics. I've not got my camera on me, but you hear me. Yeah. So that's been good.
So you would recommend doing a PhD on this topic?
Yeah, I think so. I think the nicest thing about cold atoms is maybe what Jake was saying earlier about how much control you can have. So it allows you to be very creative with the kinds of experiments you can plan and do. And it's like, as opposed to like high energy physics, you just build bigger and bigger colliders and hopefully find some new physics. Whereas in condensed matter, you kind of bring your own creativity to it and say, oh, what happens if I arrange my atoms in a lattice like this, with tuned the interactions to be in a certain way that people haven't thought of before. And then you could go to the lab and, with some effort, you can control all the variables. And
In terms of your split between like doing physical lab work and then like data analysis and stuff like that, like how is your time split generally, and like reading?
yeah, that's a good question. I guess it's different again now that we have COVID. So like access to the lab can be somewhat complicated, but in general, I would probably be spending about depends on the week, but probably about half and half on average. So half the time would be setting up things, or so most of the last year I've been working on the microscope objective and getting that working. But that's also been a lot of computer programming and thinking about how we would analyze those images, or because we're actually working below the diffraction limit of our objectives, so we have to be quite clever about how we interpret the images. So you can't just say, oh, there's definitely an atom there by eye, or by putting a box around it. So yeah, there's quite a lot of mixture of, so you come away with being like a jack of all trades and master of none. You kind of have these random skills in optics and electronics, and then also programming, and occasionally some physics, as in like reading papers and trying to understand what would be an interesting thing to do with your experiment.
Thank you for that.
Forgot to mention the most important part of the lab, which is the lab snacks corner. So there's a company that we always buy optics from, and they always send you Haribos. So yeah, that's fun. Cool. I guess if there's no more, are there any more questions, or?
I guess I've got another one if that's okay.
Go for it. Yeah.
In terms of, you mentioned about like experience in masters, like where the vacuum broke. So would you say you'd done cold atom stuff before starting your PhD? Would you say, or would you say that, or would you say it's common for people to like kind of carry on in the same topic that they've done a master's project?
I think it's quite varied. So I guess I got into this, you could maybe trace this back by, I did more experimental modules in my undergrad, and I did a summer project in an optics lab, but they weren't doing cold atoms, so they were kind of looking at things called nanoparticles, and like they're kind of on the interface of physics and chemistry and biology. So, but involved lasers. So that was helpful to just get some hands-on experience there. But I know of people who have did a master's project in cold atoms theory, and then now now do like biophysics, like simulating proteins, which is kind of completely different application, but it's kind of similar skills, I guess. People move around.
Thank you.
I've got a question to throw in. If
Great, go for it.
so, like, I know that a lot of different experiments, sort of, especially with like optical tweezers and that sort of thing, they sort of have like a measure of success of like how many atoms they can simultaneously control and stuff. So like it's not the only measure of success, but like it's one of the things that like being able to control more atoms simultaneously is sort of an improvement. So like, do you have much like truck with that, if that makes sense? Like, do you like, does this experiment have like a fair amount of atoms, or is it just sort of one at a time? Or
So there's an interesting kind of so like it's more about the difference between like a single particle system and a many particle system. So in some labs they assemble arrays of atoms from individual tweezers, and so you start with an order like 100 atoms and you rearrange them or something to create your low entropy state, so something ordered and like that's interesting to study, because we don't really want to study random noise. Whereas in our lab it's more the opposite. So like all this stuff about cooling atoms very low. It's maybe more accurately it's creating very low entropy ensembles or gases of them. And that's kind of what the Bose-Einstein condensate that Simon maybe mentioned in his talk is. So it's a cloud of atoms with essentially no entropy. And so in our kind of experiments, with these optical lattices, the equivalent metric is probably the entropy per particle. So if you measure the amount of disorder in your fluorescence image and then compare that to the number of particles, that would be the same kind of idea as a tweezer experiment saying, we can control 100 by 100 arrays. Maybe that's it.
Yeah, no, it's interesting. I guess it depends on what you're doing, as like what your thing is.
Yeah.
But no, that's
It's a similar problem but approached from different angles.